Welcome to our seminar for the day. It's good to see all of you here. And we have a very special seminar for you today. We have one of our own returning to us, a graduate from 2005 of LeTourneau with a degree in chemistry. And he went off and he got his master's at Texas A&M. And then he taught high school for a few years, and then he went back to graduate school at UNT, and he's now completing his PhD in computational chemistry and pharmacology type of stuff. Okay? And he doesn't want me to tell you this, but he recently won the award for the best TA for general chemistry in all of UNT. So, that puts a lot of pressure on him, because he's got to live up to that reputation here. He's got to give the best seminar of all semester. Yes? So when it comes out to the vote at the end of the semester, where you guys vote for the best seminar, okay, I'm going to expect Pearson to come out on top. No, Pearson is fun. I taught organic chemistry to Pearson many years ago when I taught organic chemistry before Dr. Hathaway was here. And if you can imagine a physical chemist teaching organic chemistry, we had a lot of fun in those days. So it's good to visit with alum as they return. That's one of the nice things. I joke with you guys in my general chemistry class now that they'll be coming back 10 years from now and looking me up, and you all say, no way, we're coming back and see you, Dr. DeBoer. As soon as I'm done, I'm getting out of here, right? But there they always come back. So it won't take any more of John's time here, but I would really encourage you guys to spend time with us after the seminar at lunch. Yes? We'll be over in the corner cafe up in that stage area up in the top. So if you see us over there, come and join us. Pearson can tell you about the old days here before they had a student center, before they had a Belcher auditorium, when chapel was in what we now call the corner cafe. Yes? And yeah, he can tell you all kinds of fun things about the old days, and you guys can compare stories with the new days and see how much in common there is there. And maybe I might learn something too. Always hear these things. That really happened. That was what was going on in class when you were in organic chemistry there. So, all right. So let's welcome John.
Thank you for coming, guys. I know that you're required to be here, therefore your attendance is mandatory and all those joyous things, but also let me say that your attention is also very valuable. So I will do my best to respect your time and also to make things interesting. I know a lot of you out there are not chemists, or will never be a chemist, but you probably and hopefully someday will interact with one. So my goal today is to at least give you some sort of context towards that. And with that respect, let's go ahead and get things underway. So the first thing I kind of like to address is also kind of a fundamental belief, you might say. How did I actually get here? You're going to see a lot of very interesting data, I hope, today. I hope that a lot of things here actually not just pique your interest, but really cause some curiosity and show you some of the capabilities that I've been able to highlight. These are the successes. You don't get to see all the failures or all the mistakes or all the horrible simulations that just didn't work. You also don't get to hear about the days that were dark and that were just impossible to get through that class. And yes, Dr. DeBoer was an organic chemistry teacher, but seeing a 0 out of 8 on a homework assignment just never felt good. But I learned from those things, and you will learn from your failures. And in spite of them, you'll be strengthened. So just take some heart there. I know it's the end of the semester, so there's your pep talk, guys. Good luck. You'll be fine. Keep working. And if you fail, learn from it. So, that's how I got here. I failed a bunch, and I kept on working. I had a lot of people that were supporting me. Dr. DeBoer supported me. Dr. Johnson supported me. Dr. Ball supported me. Dr. Elliott. You may know these people, you may not. If you don't, get to know them. They're really good people. So, with that said, a little bit of North Texas, where I currently am. Again, we're a fairly large school, actually the third largest in the state of Texas after University of Texas and A&M. So we're in the DFW Metroplex. We've got about 36,000 students enrolled, and about 7,000 of those are graduate students. Some funding awards. Yes, there's about $32 million worth of research, upper level research funding, and about a third of that is actually in the chemistry department. So we are actually very well funded, have our own supercomputing system that I'll talk about in a little bit, but also there's a lot of really interesting research as far as silicon wafer development and coatings are concerned. And again, a couple of programs to highlight here. Again, we do have things like biology and physics and chemistry, but also there are a master's program in biomedical engineering. There's a lot of comp sci, master's and PhD work. Again, there's a whole engineering world that I'm not going to touch on. I do apologize for those of you that are engineers. So, again, some stuff about the chemistry department. We've got about 22 professors, and that doesn't include all the adjuncts, and about 117 graduate students. But some interesting things that I actually like to highlight are actually the computational resources, because that's kind of my bread and butter, and we have a very large supercomputing system, over 15,000 cores, 1.3 terabytes of RAM space that is accessible, and about 500 terabytes of disk space, and that's just for only chemistry. So we don't have to share that with anybody. But when we do have to share, we have quite a bit more beef that we can bring in the situation. The thing I like to point out here is that's 1 petabyte of cluster space, so 10 to the 15th bytes, so it's just huge and some of the fastest data transfer around. So a lot of great resources where you can actually do not just chemistry modeling, but actually a lot of other departments, even kinesiology are doing modeling there for different statistics, and I think psychology even has a couple other kinds. So yes, it's a supercomputer, but lots of fields are using those. So if you're a CS major or something like that, and you want to work on stuff like this, there are jobs for you in lots of different places. So, breakdown of computational chemistry, kind of big 3 areas that we can not only contribute as research, but also as collaborators. The first is in pure research. That basically is where you're talking about method development, trying to make things more efficient, trying to get the better equation to solve what you're dealing with. Or maybe it is a unique experiment. Say you're dealing with something like uranium, or really toxic gases, or something that's very explosive. This is where modeling can come in and actually do a lot of the grunt work and basically do pure research there. But also where we really shine is in collaborative work, where we can basically either confirm experimental results where it's a little bit unclear, or provide insight as far as the mechanism of that reaction is concerned. Again, we can solve for these energies and we can quantify what people in the lab are actually seeing, which is one of the real strengths of computational chemistry. Instead of just a white crystalline solid, you organic people that know that well, I can give you very specific details about what IR spectra you're supposed to be looking for. What is the absorption you're supposed to see? Even what's the rate of this chemical reaction? So a lot of really strengths here as far as collaboration, but also what we can do well is optimize. So if there is a set kind of chemistry that you want to use and you're using a very expensive ruthenium catalyst, can we replace that ruthenium with iron? Can we make things cheaper? Can we do it better by picking out similar electronic states that are important? Or can we kind of give you a guided direction? Can we tell you to look in a certain direction? Can we say, look in this section of the IR field? Or can we say, hey, your products are forming so quickly, you won't be able to detect them. So we can direct and guide there as well. So those are the kind of big 3 areas of computational chemistry. And my work kind of boils down to 2 specific areas, and you might hear these terms in the future. Basically, you have chemistry divided up by what do you want to solve for. We have people that are called ab initio kind of chemists that solve based upon first principles. They model the electron or its density and try and figure out all of the rest of chemistry based upon the electron and its energy. Consequently, those equations are very complicated, very expensive to use. And kind of to illustrate this, you actually see what's called a potential energy surface there. And I don't know if y'all can see it as well. Maybe we can adjust the lights here if someone knows how to do that better. But you can see this little kind of ball, kind of, as I'm nervously twitching, kind of trying to get up over this hill. Again, it's a reaction diagram. So as there's energy flowing through this system, maybe you're giving it too much vibrational energy, and therefore it's not actually able to have enough energy to do this reaction. Or if you give it too much energy, it actually goes on a very, very similar straight trajectory every single time. So we can only solve those kinds of questions by figuring out where those electrons are. Second is kind of with the little bouncing molecules up top there. Those are basically classical methodologies where you're using Newtonian laws, Newtonian physics, kind of charge, radius, or mass charge forces to solve for what the activity of that molecule is. And in small scale systems, it's much, much cheaper because you're talking about a mass, you're talking about basically sums instead of integrals. Triple, third integrals, you're talking about, again, just a whole massive Taylor expansion instead of something like a partial derivative. Okay? So easier math on top, more complicated math on bottom. And to that end, we also have basically multi-scale modeling, where we have a whole bunch of different systems here. We start down at the bottom with our ab initio methods. Again, we can only do this for anywhere from 10, maybe 20, maybe 30 if you're pushing it, kind of methods to solve where all those electrons are, where all those orbitals and how they're occupied, to larger systems where now we're talking about our density. Here you can go anywhere from 10 to 20 to 100 atoms. And again, those are what are called density functional theories. So again, density based upon those simulations. And then as we move to even larger, not only is our time scale kind of going up, in other words, these simulations are instantaneous, there's no dynamics here. These simulations, again, you get to see the movement, but time is not a big issue. As you start to increase the size of your systems, you need to basically increase your window of what you're observing. So as you're talking about one of these large systems that has anywhere from several thousand to several hundred thousand atoms, I'll be talking about grand canonical Monte Carlo stuff today. But also when you're talking about millions, if not billions of atoms simulated with critical fluid dynamics or coarse grain dynamics, here you're treating whole sets of molecules. I don't know if you can see it very well, but these are all little individual spheres with their own individual parameters, but they're simulating giant hydrocarbon chains. Okay? So depending upon what you're looking at, you need to either change your method to make it basically a simpler method or a more complicated method, or change the window of what you're observing it from instant, no second deviation here, to, in some cases, tens of seconds, which is just insane to me because that is so much data. But people do it, and they work. So what am I going to be talking about now that the introductory material is kind of done and out of the way? Three things that I'm going to be discussing today are nitric oxide activation. So in the enzymes in the body, there is a signaling molecule called nitric oxide. I'll talk about why that's important and what we hope to be doing with it. I'm going to be discussing metal organic frameworks, a very interesting and novel crystalline material that hopefully you may have seen reading even Time magazine. Okay, it's very, very popular. It's basically the next generation of all those nanomaterials. Really cool stuff. Growing field for material science, especially. And then lastly, I'm going to give you a little bit of an example of what I do. Actually, I am a UNT student, but I also work for Reata Pharmaceuticals. I basically do exploratory research for them. I tell them which drug molecules work, which ones don't, and why. And I'm going to give you all an example of that with this model of S-kinase 1, or S6 kinase 1. So unfortunately, I can't talk about any of that work that I did for private sector, but it's close enough. You guys are okay with not exact, copyright material, right? So, to continue, enzymatic activation of nitric oxide. Why is this important? What's the purpose of this project? It goes back to actually combustion. All of y'all are probably very familiar with the idea that hydrocarbons produce pollution. Okay? Things like nitric oxide, the molecule I'm talking about, but also several other molecules, sulfur dioxide, carbon dioxide, all these other kind of dangerous gases that, if left in the atmosphere, cause changes. Specifically in 1996, the Clean Air Act actually said that power plants needed to reduce the emission of sulfur dioxide, but also nitric oxide. And you can kind of see here from the projection of power plant emissions going from anywhere from 7 or so thousands of short tons now to a predicted model of basically close to under 2,000 short tons per year. And the reason why that is there's a transition from coal-based kind of energy generation to natural gas, or to solar, or to nuclear. So you have all these other options with your power plants. But a power plant is very, very different than what goes inside your car. They're talking about high temperature, high pressure outputs where you can concentrate gases and make them much more viable. In your muffler, you don't have high temperature, relatively. You don't have high pressures. So you have to figure out a system that will actually work to activate those same pollutants. And that's kind of where biology comes in. Because biology works at room temperature and pressure. Your bodies are doing all this stuff right now nearly instantaneously. And what's great about our nitric oxide system is it is very, very well studied. Again, if any of you have had biochemistry, you might loathe things like this, but the important thing I want you to realize is that nitric oxide is actually basically what is a signaling molecule that, yes, it has this whole cascade of activity that follows it, but it controls lots of things. And because it controls lots of those signals, it's linked to lots of different diseases. Diabetes, lots of different forms of cancer, hypertension, migraines, meningitis. All these have had physical links to nitric oxide. So biology has dedicated a lot of time, energy, and resources to learning of that. It's produced from arginine. I shouldn't say several, but come on. There we go. Back in 1998, Nobel Prize was awarded for nitric oxide research. And you can see the kind of surge in papers here. So pre-Nobel Prize, it was doing, you know, several hundred, several thousand kind of papers. And once the Nobel Prize awarded, lots of people, again, some more money, more time, more energy. Again, this is in physiology and medicine. But as a chemist, what I can do is then take this information and then utilize it through data mining to talk about that reaction. Because the chemistry that's occurring in an enzyme is still the chemistry I want to have occur maybe in your muffler. Okay? So the way we start with this is we started by looking at over 100,000 protein crystal structures. What these are highly resolved, accurate, three-dimensional structures of proteins. Their amino acid sequence, but also their three-dimensional representation. So here we kind of have an example. Again, it's not just all letters. Each one of these letters symbolizes an amino acid pair. Sorry, not pair, not DNA. These are all amino acid side chains. And then what we can do is overlap and overlay complete proteins. So we had to actually pare this down quite a bit, going from 100,000 to only 98, to make sure that we have a full protein. Because if I just have a little chunk of it, yeah, it might overlap, but it doesn't describe that full enzymatic activity. So by getting the full enzymatic activity, we first started by doing what's called sequence alignment, where it's just as simple as it says. The optimization of it is a little bit more complicated. But it starts by just aligning. You have a threonine, you have a threonine, you have a threonine, all lined up, good to go. There are a couple other things that happen. These gaps actually can form where you have weaknesses in the protein structure and whatnot that are still allowed to a point. But what it does tell you is how similar all these proteins are based upon not only just that amino acid kind of first layer of information, but the second layer of information where you have that structure. Okay? It's similar like in GenChem where you have C6H7 or something like that, and you can arrange that lots of different methods. When you actually know that structure, then you can talk about what that actually is. So it's a protonated benzene, in my brain at least. Who knows what other people are thinking. So from those structures, we actually sorted out and grouped them based upon how those enzymes bound nitric oxide. And you can kind of see here, I know it's a little blurry, I do apologize, but right there in the center of this enzyme is nitric oxide. It is bound to, in that case, copper, and then there are 3 histidine amino acids around it. So we grouped them based upon just how is nitric oxide crystallized in this protein. You can see most of them are based on hemes. Hemes are similar to what are in your hemoglobin, actually in some cases identical to. So nice big flat structures that have an iron kind of core. And you kind of see these similarities here where we do have all these kind of flat structures with a little red and blue guys sticking up there. Where these are all different though is how you have different amino acids, again, kind of underneath, where you have here, you've got a valine, here you've got a tyrosine, but you've got some other methods that are also very, very different. You've got the histidine model, but then you have these weird sandwich complexes, you have iron-sulfur clusters. So all this diversity, basically, you need to deal with it, because not all 51 of these structures that have basically this heme are going to be exactly the same. So the way we do that is actually basically establishing them into subgroups, different motifs. Basically means different three-dimensional arrangement of those guys. So we have actually 5 different groups. All 5 of these proteins have nitric oxide bound to an iron heme that has a histidine trans to it. So when we took all those 51 groups and lined them up, did that sequence alignment, you can see kind of a pretty quiltwork pattern that doesn't actually tell us anything. When we remove the extraneous data and say that these proteins do not align, they don't have good overlap, we then kind of condense it down to 5 different subgroups based upon that overlap. So a real methodical way of defining these groups. So where you have high overlap, then you're in a group. If you do not have overlap, then you're not in those subgroups. Again, the whole purpose here is through homology, through comparison, you can determine what are the important features of a chemical reaction. So again, final grouping here, this is what we started with, some 98, and then following the kind of filtration, again, whenever you're doing data mining, just like in real life, mining, you have a lot of slag to remove and you're only trying to get that nugget. So these are the nuggets, you might say. So here we have all of our aligned models. Again, you don't really need to remember the names and the individual groups, but know that they are similar in only one respect, how they bind nitric oxide. Even though this might have a histidine and it be part of a heme, it looks very, very different to this group. So you have all these diverse proteins that are essentially doing the same job. Whatever they do that is the same is critical to the reaction. If they don't do something the same, then it's not. It's just that simple. Again, low-level theory for lots and lots of data. we're going to increase our level of theory and actually deal with smaller amounts of data soon. So first thing we looked at was the geometry, or basically the bond angle and bond distance of our active site here. So here you can see basically the long list of different groups and how their bond distance going from our metal center to our nitrogen. All of them did connect at nitrogen except for actually that copper group, which I will talk about. A little bit different arrangement there. And you can see that in the bond angle. Here, these are anywhere from 120 to 150, so anywhere from kind of an angle here is what I'm talking about. I know the picture looks linear, but really, trust me, there's a bend there. So we have similar geometries, but not quite identical. So the geometry that you might need for an iron system, yeah, that's highly conserved, but for a copper system, it might actually need to be something different. So that's a consideration. What's that optimal geometry? What's that optimal distance? The next thing we looked at is when you're talking about an enzyme, you're not just talking about that one center where things are happening. There's an environment around this area that is called the pocket. The amino acids that either help shuttle the molecules in, maybe help kind of hold everything together as far as the structure is concerned. And we kind of see here, we've got a kind of outline. This is basically a magnification of that. And the amino acids that I've highlighted here are kind of in the circles and sticks kind of arrangement, where you can see these are the amino acids that are basically going to influence that bonding event. So those are the ones that we want to really focus in and learn more about at a higher level of theory. So we're ignoring the rest of the protein simply because it's not going to influence this event. So the first thing we looked at again, this is the pocket region, is the pKa. And some of y'all remember our pKa is ratio of protonated to deprotonated acids. In proteins, it's a little bit more complicated. It still fundamentally means the same, but we have to take a few other things into account because pKas can actually deviate from what you might look up in, say, a nice big manual. So depending upon whether there's an intrinsic, so this is our starting, what you would look up in a manual or a CRC or some sort of database. And then there's all these other things. Is there desolvation? Are there water molecules around? Is there hydrogen bonding? Is it actually a charge separated type species? So in other words, different parameters for proteins for specific pKas. Higher level of theory, less atoms in our system. And as we have here on the graph, the pKa here is majority neutral. Some slightly acidic, but again, right around the average pH of the body, which is about 7.4. So again, this basically tells us that we have a neutral to slightly acidic system. So high basic systems would not be applicable, which is great because as you're burning stuff, you have CO2, and that's going to be forming carbonic acid. So the fact that we do have some of these that are slightly acidic means that hopefully our enzymatic process isn't as sensitive to weaker acids. Next thing that we looked at is actually something called hydropathy, or you might know this as lipo or hydrophilicity. Basically, how attractive is that water molecule? The way we do this is talk about the energy basically associated with those amino acids and what happens when you solvate them and then desolvate them. And some of you might recognize the kind of chemical equation here where we've got basically our partition function and our fugacity that's basically talking about what's the ratio of that to the surroundings and also to the compressibility. But what does this actually mean? If we take our protein model again, we're looking at just the amino acids in this region, and let's color code it, make life a little bit easier. We can produce some pretty pictures where we have basically what are hydrophilic and lipophilic. So if you dissolve the green sections in water, they're going to actually have a positive energy, as indicated on this graph. Most of our proteins actually take some energy to dissolve. They take some energy to have water molecules present in this kind of pocket region. And as you can see, most of it is the lipophilic, which means you are fat soluble, and sometimes you'll hear enzyme type people call this a greasy pocket. Proteins, again, they are greasy. They have pockets that will squeegee out water. It's great. But is water important or not important for our system nitric oxide? That's the question we're trying to answer. And our answer here is maybe, but probably not. Again, the majority of these will, in fact, have water expelled from that pocket region. But we do have a couple that are either close to zero or, as you can see with our one example, and that's actually a large system where we have copper and the heme-histidine complex, and that is actually solvent exposed. So water in the system, possibly, but most likely not an issue. All right, now we're going to our next level of theory. We're going DFT now, density functionals, but we're talking about smaller and smaller systems. So in those pockets, you have different amino acids. Some of those amino acids will influence the bonding event. Sometimes they won't. Even though all the amino acids might have something like a valine amino acid, that might not interact with nitric oxide. So we're talking about which amino acids are critical to that binding event now. So the way we did this was actually with quantum mechanics, using basically those methods that we talked about for solving exactly where those electrons are and their energy. And we have two different methods. We have functional methods here, and also we have ab initio methods. So again, two levels of theory, similar statements, similar energies that we're looking at, but the main reason there is, let's get a complete picture. So the first amino acids that we looked at here were all conserved in the pocket. Some of them are going to have basically a negative binding enthalpy, which means that they release energy when they're bound to nitric oxide. So again, don't get caught up too much in the fact that not everything is highly conserved. What I want you to kind of take home from this graph is that things like histidine, glutamine, arginine have anywhere from 10 in some models, but in some models all the way up to 25 or 30 kilocalories per mole of binding. These are small, charged side groups, like glutamine and like glycine, that don't get in the way of it bonding to the backbone. Whereas other groups, like serine, they're neutral, like tyrosine, that is also neutral, do not bind it as well. So when we model those reactions, we need to be sure to include things like arginine, things like tryptophan, not so much. So it helps us know and discriminate which amino acids are critical. Secondly, we looked at different analogs. And it might not show up as well here, but there's our nitric oxide, and here's our sodium, a nice little sodium cation. Again, this isn't necessarily similar to any amino acid, but sodium is in your body. It will influence binding. And I know it's a little bit difficult to see here, but this is our benzene ring, again with a alcohol, so this is actually phenol. And that's really similar to an amino acid called tyrosine. So modeling this to make sure that we are consistent, that yes, small charged things, like the glutamine, but also like the sodium that we have there, have a very high affinity for nitric oxide. So they can influence that metal bonding. And then the next place where we're going to take this is actually describing that metal bonding event, and that's currently what I'm actually working on. So, won't get to talk about that today, but a quick little summary. Some things that are important. pKa is important. You need to have those small charged residues present in future simulations. But neutral, not as important. Water, not as important. So, with that, we'll conclude our nitric oxide portion and move on to metal organic frameworks. So those of you that are very interested in material science, about novel materials, wake up, here's your stuff now, if you want to learn about something kind of cool. So these are metal organic frameworks. It's very similar to, and I might be older than you all to get this reference, but Tinker Toys, where you have basically a node and a linker. You have basically, typically a metal node, kind of the corner piece, and then the linker can be lots and lots of different organic molecules. So what you basically have a chance here is a customizable framework that has all these cool little pores. It has all these openings, and each one of those openings can be unique to the models maybe you want to capture, maybe you want to store, maybe you want to activate for a catalyst, maybe you just want to filter out. Okay? So it's basically the next level of nanotechnology, because it's using chemistry that's actually established in some cases hundreds of years ago to do these reactions. But then we take those and couple them and make a much more diverse, much more customizable system. So the MOF that I was working with was called fluorinated MOF1, and you can kind of see a picture here. It kind of makes a hexagon. And all these spheres that you see are actually toluene, or sorry, those are nitrogen atoms. So the pores are actually quite large. And what's really interesting about the fluorinated MOF that I worked with is it actually changed size as you change the temperature. So you basically have a MOF that can expand, maybe when you're loading it with CO2 or oxygen, and then contract when it's in storage mode. And then when you need to release it, it can expand again, just by changing temperature. So really a novel system. And so some of the stuff that I was trying to explain, and again, we're going to our theory again, just a little bit. I tried to limit some of the equations. But these are the big important ones. Again, what we're modeling here are actually things like bonds, angles, and torsions. You're basically treating this like a spring in some cases. In some cases, you're treating it like a point charge, where you've got basically positive, negative, being attractive. You've also got where you have two positives, and this is basically a polarizability. What happens if you have two positives next to each other? Are going they to slightly repel? How much of this, when it's connected to the rest of the system, is going to change energetically? We also have van der Waals forces. As you have these dispersion corrections, how is that going to change? And what's nice about Monte Carlo-based theory, and force fields specifically, is all of these are parameters that basically you don't have to solve integrals for. Again, it's a Taylor expansion. It's just a sum. It's pretty straightforward. So we're not solving quantum mechanics here. We're doing classical Newtonian stuff. So these are all run basically in serial. You only need one processor. everything else that I talked about earlier was all run in massively parallel, 16 cores, in some cases 32 cores, with anywhere from 500 gigs of memory that are required to, in some cases, several thousand. Okay? So much, much cheaper calculations. And we can still do quite a bit. The first thing I'm going to talk about is actually how do we model this. And we have two different systems. We have our framework here. This is our MOF framework, where we have silver atoms kind of caging a pyrolyl system that has these fluorinated carbons at the end. And then, sorry, this is static. This doesn't move in our simulations. I know I said it was breathable. We don't quite have the capabilities to make that breathable just yet, but we can model each step and kind of simulate that breathing. So that is a static system, and that's been characterized by a generic MOF force field that has those charges that we talked about earlier. And then we actually have our guest molecules. Here are our nitrogens kind of hanging out in that pore. And what's interesting about the models here is those are not actually behaving like ideal gases. They're not behaving like liquids. There's an equilibrium that is occurring that is basically not only based on the attractive forces of that structure, but also kind of the own properties of that liquid gas kind of equilibrium that's occurring. So that's actually been accounted for in what is called a transferable potential four-phase equilibrium. So you're modeling this as a gas, as a liquid, somewhere in between, which is important because it's not behaving nearly at all like an ideal gas. So a couple of things with Monte Carlo theory. Some of you might be familiar with it. It's that idea of basically a randomized sample set that then eventually will achieve an average. So there's a couple of ways that you can get that randomized sample set, a couple of different kind of choices that are made by the system based upon energy. So if you kind of limit some of the things, like how much energy is in the system, that's actually going to be what's called an initialization step, where you're allowing the number of molecules to change, but the energy of the system is not going to change. So you're not actually doing full Monte Carlo methods here, full grand canonical, but it lets you achieve the right number of molecules. And then when you do, introduce the energy into the system, this is now our actual system cycle. So again, the main idea here is you're populating your box first, and once you populate the box, then you can actually talk about the energies that are associated with it. And then the second thing to be concerned with, after you basically primed the pump, as it were, is how long do you run that pump? How long do you run those steps? This is actually what's called a helium void. It's that empty space that is in that MOF. And what we're doing here is basically inserting and removing a helium particle, atom. And as that successful or not successful insertion occurs, it then gets summed into an average. It's great, sums. But over time, you're going to see basically a set number of that average kind of start to flatline, even start to behave slightly periodically. So at a point in my simulations, I chose kind of 50,000 based upon that we're varying, again, those are our error bars. We're within error for those number of steps. So that's kind of how we came up with some of the parameters that we generated for this. You'll hear some people doing simulations for millions of steps. Again, because it's an average, if you don't have a good initial guess, then you don't really know where your average is supposed to be, therefore you need more steps. If you know about what it's supposed to be, then you can get away with fewer. So, back to our breathable MOF. So we were fortunate enough to have experimental collaborators that had crystal structures at lots of different temperatures for this FMOF1. So we wanted to be able to model these helium void calculations based upon the experimental voids. So again, we have all these different crystal structures that we were to run helium void simulations with, and we can see that our helium void fraction results and our experimental results are actually pretty close to alignment here. We're about in that 100th place off. So as far as a static physical component, spot on. It means our simulation, at least in this part, works very well. Where it might not perform as well is in our next area. Here we're actually talking about uptake of methane. So instead of that helium particle being inserted and removed, now we're talking about CH4. Again, it's the same MOF system. We have it at multiple temperatures, multiple pressures, and we're trying to match our experimental data for uptake of methane. So we've got basically a volume-to-volume ratio saying this is our volume of our guest gas, the volume of our framework, in this case FMOF1, and then basically what is that ratio of the two. And as you go higher, higher, higher on that volume and volume ratio, that means that low pressures, and as you increase the pressure, you're going to actually have increased uptake. And again, this is the lowest temperature. So high temperature, higher pressure, more uptake. That's more due to just the overwhelming idea of basically chemistry. You've got dispersion, you've got, word dropped? Oh well, y'all won't remember that part. But the important thing from this graph is grand canonical Monte Carlo stepping up to the experiment. And this isn't my brightest point here. We actually are overstepping our estimates in some cases as much as 50% of that volume to volume ratio. So what's going on here? Why do we model our helium so well, but here our methane uptake not nearly as accurate? Mainly what's going on here is somewhat the experiment, the way it works, is basically a uniform gas is populated, and then the mass of that system is basically determined on that layer that occurs. So you can get a mass, you can figure out how many nitrogen or whatever atoms are in there, and then based upon that mass, you can talk about that uptake. The experimental system is not ideal. It has lots of surface breaks, there are lots of issues, or even they're not getting proper uptake all the way. So some of this, there might be some experimental error. Maybe we can talk about that for some of the lower pressure, higher temperature models. But some of the models, it just doesn't model well because as you're going to higher and higher temperatures, you're dealing with more and more energy. So low temperature, low pressures, this method might be very effective. But as you go to higher and higher systems, because you're increasing your energy, you need to increase how you're actually parameterizing this. So yeah, lower sections okay, higher not as much. But what we can do is, because we are overestimating at all the different areas, we can make a comparison. So even though it might not match experimental data, we can make a comparison against other models to see how it competes against them. So here we're looking at 4 different uptakes of hydrogen, nitrogen, oxygen, and CO2. And again, it's the same idea here, but you'll see that we're talking about very low pressures. So this is -5 log of the, actually that's in bar, I believe. So very low pressures, and temperature here all in Kelvin, so fairly low temperatures. So what we're looking at here is which one of these performs the best at the same conditions. So if we're trying to tell our experimental collaborators which models they should probably pursue, we can definitely talk about how nitrogen, oxygen, and CO2 are very important, but maybe not go for the helium models. The design of this MOF is not ideal for that. And we can back this up with, we're going back now to quantum mechanics. We can't model the entire MOF, but what we can do is truncate it. Again, this is with density functional theory, so we solve for the density of the electron wave function for all these different atoms. And our potential binding energy, again, very clearly shows oxygen has the greatest amount of binding energy. So this graph actually mirrors what our uptake data shows, even though these methods were solved on a single core, maybe 2 gigs of RAM, and this took about 2 weeks with about 32 cores and 500 gigs of RAM. Okay? So we might not be able to address all of our answers, but we can get some of the trends approximately correct. So, in conclusion for our MOF section, some of the important things, we can do physical volumes and physical properties quite well. Uptake, absorption, not as much for independent kind of pure determination, but good for comparisons. And lastly, based upon this evidence, we can talk about oxygen having very good uptake over even nitrogen and CO2. And I could get into which orbitals. It's normally the F orbitals from the fluor, or sorry, the P orbitals from the fluorines, but we have more information with those as well. So we're not able to model the dynamics of this basically expanding just yet, but we can model it. It's metal organic frameworks. Again, they are a customizable surface material, crystalline material that you can do lots and lots of powerful chemistry, material science, mechanical properties with. Okay, novel field that's expanding all over the place right now. And finally, inhibition with drugs. You can sometimes make really good comparisons and give people really good directions to go based upon a simple comparison, based upon an idea that you have. Again, I've learned so much from Dr. Bohr, Dr. Johnson, but also I've learned a lot from my current professor, Dr. Angela Wilson. She has been a great mentor, a great advisor, and if you ever see her at a public talk or something, she has giant, poofy, blonde hair. She's very noticeable, but also she's very, very smart. Leader in her field, one of the premier computational chemists currently. And again, I also have to thank Dr. Kandari. He was very influential in the pharmaceutical and worked with Riata, and also our experimental collaborators, the Omari group, who actually made those MOFs that I was dealing with. It was a great collaboration, continues to be. Funding came from the Department of Energy and NSF funds, and also Riata pays my checks, so I got to thank them as well too. And thank you for your time and attention. I'll do my best to address any questions.
So you're still holding that 0 out of 8 against me after all these years.
I earned it. I earned it. And what was worse is it was in crayon. When his girls were little, he would make them grade in crayon. And it was just so disheartening to have like a 4-year-old say, "You got this wrong." It's just
No, I stopped doing that, I just let them do the happy faces after that.
Oh,
Because
okay, thank you.
But anyway, other questions for Pearson besides, oh, I graded this paper back a little bit.
You were better at PCAM.
Yeah.
Much better.
Oh, yeah. Well, becomes a better subject again. Yeah.
It has its strengths.
All right. Questions though about computational.
Oh, I didn't even get to talk about my cluster construction. Another project that's not even up here is actually I was able to build a 200 core cluster with surplus library computers. All this stuff is free open source software. It's basically a miniature supercomputer out of spare parts. It costs like $1,000 to fix. It was great. I learned so much stuff, and pseudo privileges are fantastic, by the way, if you are people that know what pseudo actually means. It's really fun to make people think that their files always temporarily.
So, questions for Pearson here.
Oh, I was on dorm 4, by the way. Any dorm 4?
Dr. Paul has a question.
Here we go. All right.
John, we had a little bit of a pipeline of students going to UNT.
You
We had
did.
some summer students who've done research during the summer.
Ben Hobb actually used the cluster that I built. And Dave Coots is another admin that, if you guys know him from his newspaper days.
Back there.
Yeah, Dave is still up to his nefarious acts of,
Same shirt he had on when he was here.
if any don't remember or know of Dave Coots, he is a fantastic individual. He's actually the guy that I share an office with, so he's still plugging away. And I still actually have some scripts by Tom Grimes that I occasionally look at. So yeah, there is a Laterno presence in our group and nearby, and it's good. It makes a difference on who you work with, who you share intimate space with. Sometimes when you get frustrated, when you are, oh, I don't want to study anymore. It's nice to be with people that you share a background with.
We would like to keep that pipeline going. So,
Well, again, summer research programs are fantastic. We always are accepting students, and especially if you get a hold of me, I'm sure Dr. DeBoer will be more than happy to send my contact information, and I'll be more than happy to. I'll even take you on if I'm allowed to. So, see if I can manage and feed all the dogs. But yeah, get some research while you can, guys. It opens your mind.
Okay. Well, we can officially close and let John entertain questions up front. Also, we'll be over in the corner cafe, and we'll be around for a while.
If
We
you're
have some,
chemistry people, you want to
stuff.
learn about graduate school,
Yep, we got-
UNT stuff.